Okay, Vandana, hopefully you are feeling good and you are able to conduct this interview and feeling comfortable. Okay,
Yes.
so if you want to take some time, then you can.
Okay, sir.
Okay, so be normal and be confident, and then we will start. Have you discussed someone else who have appeared in this interview?
Yes, sir. There are a few friends I have talked to regarding this interview. They have given me reviews regarding their interviews. Though they said they were asking questions from majorly from our subject biotechnology and some IPR questions as well.
Okay, and also I will suggest you to just watch the Himanshu's experience of real interview videos. You will learn so many things to that video
Okay, sir.
because so many students have their curiosity that which topic and what the area will be prepared for that. But in the last of this interview, I will also suggest to you what will you have to do. So we start. So Vandana, just introduce yourself in brief, so we can start further.
Yes, sir. Good evening, sir. I'm Vandana Gautam. I'm 22 years old from Delhi. I have done my schooling from Delhi as well. I have done my bachelor's in sciences from Acharya Narendra Dev College, University of Delhi, and my master's in biotechnology from Guru Nanak Dev University, Amritsar. From there, I have done my dissertation thesis project regarding the approach for early detection of Alzheimer's diseases, and my interests include traveling, acting, and dance.
Okay, if I ask your area of interest and favorite subject, then what is your?
Yes, sir. So
Yeah.
my area of interest is neurosciences, for which I've taken up that project as well. And my subject of interest are immunology and molecular biology and plant biotech as well.
Okay, Vandana, tell me, antibody has two major function. That is,
Yes, sir.
one is the binding to the antigens and perform an effector functions. So, which part of antibody or which terminal of antibody has ability to perform the effector role as well as binding role?
Yes, sir. So the epitope of antibody is majorly involved in the binding towards the antigen.
Okay, and for the effector function,
For the effector function, so the he- heavy chains.
there is a specific term for that. Antibody have a two regions.
Yes, sir. Heavy chains and light chains.
No, no, this is the part. This is the peptide chain.
Yes, sir.
Have an antibody have a two that is fragment, two part. Recalling you, one is okay. Don't worry. It's okay. It's a fine. Vandana, in antibody, how many level of a protein structures are formed to complete a structure of antibody? That is primary, secondary, tertiary, or quaternary. Which kind of structures of the proteins are involved to form a antibody?
Sir, secondary and tertiary structures.
Secondary antibody.
Yes, sir.
Can you explain what is the meaning of secondary antibody?
No, sir, secondary structures of proteins.
Okay, secondary structures. So, can you
Yes,
explain
sir.
what is the secondary structure of the protein?
So secondary structure of proteins generally includes the beta sheet and sorry, sir, I'm not able to recall this.
Okay.
That
So, have you ever
particular name.
seen that the any proteins are functional in their secondary structures or primary structures? Have you ever seen?
Sir, I'm not aware of that.
Okay.
Mm.
Any proteins of not specify the antibody. Any proteins in our body are functional in which structure of the protein, in which form of the protein, either primary, secondary, tertiary, or quaternary.
Sir, I think tertiary and quaternary
Tertiary and
structures.
quaternary. Can you give some any
Yes,
examples of a tertiary and quaternary structure of the protein?
yes, sir. Sir, glycoproteins.
Glycoproteins are the just chemical composition. This is not the protein.
Yes, sir.
I am asking about the
Yes,
name
sir.
of the protein. Any examples of a tertiary or any example of a quaternary protein?
Chaperones are there. Uh,
Chaperones. Chaperones are the folding proteins. They
Mm,
are involved in the protein
yes, sir.
folding.
Sir, it is involved yes, sir.
Okay, chaperone proteins are what? That is tertiary or quaternary?
But I'm not sure about that.
Okay, okay, leave it. Uh, Vandana, for determination of antigen, antibody have a specific reason,
Yes, sir.
right? So can you tell me the properties of that specific region with the name, with their name?
So the paratope region is that determines the antibody.
Paratope.
Sorry, the antigen. Yes, sir. So paratope region of antibody interacts with the epitope region of the antigen.
So what is the CDR? Have you heard about this?
So complementary determining regions.
What
Yes.
is this?
So these are the regions that. Mm,
I think you are feeling so nervous, and right now you are not comfortable.
yes, sir.
Okay,
Yes, sir.
so if you want to take some time, then you can take and be relaxed. Then we will continue. You have to appear for the main real interview for next tomorrow, no?
Yes, sir. Yes, sir.
Okay, so don't feel bad. Okay, just calm down and be comfortable. Okay, so my question was that what is the CDR?
So I'm not able to recall.
Okay, 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 okay. So on the basis of light or heavy chain, how many different kinds of antibodies are there in the human body?
So there are two types of antibodies, majorly polyclonal and monoclonal antibodies. Um, yes, and the classes of antibodies are total five, which are the IgG and IgA, IgM, IgE, and IgD antibodies.
Among these five different kinds classes of antibody, which antibody have highest serum concentration? That
So
means
IgA,
IgA.
yes, sir.
Are you sure?
Yes, sir.
I am asking about the concentration of antibody in the blood. Among these five different kinds of antibody, which antibody has highest concentration in the blood?
So I'm not sure, but I think it's IgM. IgM.
IgM. Okay. Uh, a single antibody can bind how many different kinds of antigens?
A single antibody can bind two types.
Only two. If I
Yes,
if
sir.
I ask about the antibody M, then what is the number of what will be the number of antigens that can bind?
So as antibody IgM is pentavalent, so it will bind to 10 epitopes,
Okay,
as it is bivalent in nature and
okay, okay, got
sorry,
it.
two binding sites
Vandana,
in pentavalent.
Vandana, suppose you are doing a gel electrophoresis, right?
Yes, sir.
And you have an antibody that is antibody A, and you have to run SDS phase electrophoresis. So,
Yes, sir.
what will be the number of bands on the gel? Suppose you
So
are,
number of
yeah,
bands.
yeah, number of bands. You are dealing with antibody A.
Yes, sir.
Yeah,
So it is antibody A is bivalent in nature. So I think there will be two bands of different sizes.
only two bands. Have you heard about the J chain?
Sorry, sir.
J chain, J chain,
No,
J
sir.
for jackal, J chain.
No, no, sir.
Okay, if I the same question for the antibody M, then what will the answer? If you run electrophoresis for the antibody M, then what will
Yes,
be
sir.
the number of bands on the gel?
So five maybe.
Five. Have you idea about the chemical composition of antibody? How many different kinds of polypeptides are there in the antibody?
Yes, sir.
Okay, have you heard about the light chain or heavy chain?
Yes, sir.
So how many number of a heavy chain or how many number of light chain are there in a single antibody?
So four heavy chain and two light chain.
Four heavy chain and four.
Light chain.
It means it's a
Sorry, sir. Four, two variable heavy and two light, two variable light chains and two variable light chain regions. So
Then
four
what were the
four
constant
heavy
region?
and two light.
There are also constant region in antibody.
Yes, sir.
So how many number of peptides are there in the antibody? If I ask a single antibody contain how many number of peptide chain?
Sir, mm, I
No
have no
idea.
idea about it.
Okay. Can you tell the can you just suggest me the different kinds of a chemical bond involved in a antibody formation? How many different kinds of chemical bond are there?
So there are the light and heavy chains are connected with disulfide bonds, which contains cysteine amino acids, and the antibody together are bind with a peptide bond.
Sorry, antibody together with
So peptide bond.
okay. Peptide bonds are found with what? Antibody,
No, sir, amino acids
amino
to
acids.
the
Okay.
protein. Yes,
So there are only two bonds, that is disulfide and peptide bond. Any other?
yes, sir.
Any other?
Uh, no, I'm not able to recall the other one. Other.
Okay. For the formation of a tertiary structure of any protein, which chemical
Yes, sir.
bonds play crucial and important role?
For the formation of.
For formation of a tertiary structure of any protein, not dealing with
Yes,
the
sir.
antibody, any kind of a proteins, which chemical bond will play crucial and most important role?
So hydrogen bond.
Hydrophobic,
Hydrogen bond.
hydrogen bond,
Yes, sir. Non-covalent majorly.
non-covalent majorly. Uh, can you suggest some name of a non-covalent chemical bond? Type of a non-covalent
So
chemical bond?
non-covalent, yes, sir. Hydrogen bond is there. Hydrophobic bond. Van der Waals.
Hydrogen bond is a non-covalent bond. It is a bond.
Yes, sir.
Hydrogen bond is a bond.
So hydrogen bond is a generally a bond present in the water molecules.
I am asking to you, it is hydrogen bond is a real bond.
Yes, sir.
A bond can be defined at what? How you can define a bond? A chemical
A
bond is
bond
what?
is defined as yes, sir, that connects two molecules.
Connect two molecules.
Yes, sir.
Okay, so hydrogen bonds can connect two molecules.
Yes, sir. So the oxygen of the in water molecules there
Okay,
hydrogen bond
Vanna,
is
tell
present.
me, is there any criteria for to form hydrogen bond, or each and every kinds of elements can form hydrogen bond, or there are some specific elements having ability to form the hydrogen bond?
So there are specific elements that form hydrogen bond.
Can you say the name of that specific element?
So water is there.
Water is not an element. Water is a molecule.
Yes, sir. Molecule. Yes.
You are. I am asking about the element. Suppose I provide you five different elements like the carbon, phosphorus, potassium, calcium, sodium. So among these different type of element, which element will form hydrogen bond?
I'm not sure, sir.
Not sure. Okay.
Yes,
Okay.
sir.
Okay. Just relax. Don't worry. So, as you stated in your form that you are interested in the immunology, so that's why I am asking majority of
Yes,
the
sir.
questions from the immunology. But if you feel some comfortable in other subject, you can just inform me. Then I will ask s- different questions. Have you understand the genetics,
So genetics and molecular biology
molecular biology,
we can do and plant biotech.
plant biotechnology? Okay, uh, Vandana. Nowadays, in plant biotechnology, one term is very crucial and important, that is the plant growth promoting rhizobacteria, pgpr. Have you heard about
Yes,
this?
sir. Plant growth promoting rhizobacterium.
How nowadays plant growth promoting bacteria draw the attention of all the researchers in the plant biology and soil biology or agriculture biology. Can you suggest some name of the pgpr bacteria, that is plant growth promoting rhizobacteria? Just few name.
So the agrobacterium rhizogenes is there.
Agrobacterium is a plant growth promoting rhizobacteria.
Yes, sir. Sir, in a way, it promotes the production of secondary metabolites upon the hairy root disease.
Okay, how PGPR can promote the growth of the plant? What are the basic mechanism? How a rhizosphere bacteria can promote the growth of the plant? What is the mechanism behind it?
Sir, in a way, it infects the wounded plant, which generally releases the secondary metabolites, which are essential for the protection and defense mechanism in the plant.
Okay, can you suggest some name of secondary metabolites which involve in the defense mechanism?
Sir, terpenoids and alkaloids are there, uh, and phenolic compounds.
These compounds are secreted by the plant itself, not from the bacteria. Uh, and, and,
No, sir.
and my question was about the bacterial role of the plant development. Terpenoids, alkanoids, and flavonoids compounds are the
Yes, sir.
secondary metabolites secreted by the plant itself.
Yes, sir.
That is the plant itself. They have own defense mechanism. My question was about the how PGPR promote the growth of the plant. How PGPR play the role in the growth and development of the plant.
I'm not aware of this
Okay,
concept.
okay, that's it. That's final. Uh, have you heard about the cytoplasm?
Yes, sir.
So, what is the cytoplasm and protoplasm?
So cytoplasm is defined as the area of a cell which is bound inside the cell membrane, and the protoplasm differs from the area which is bound inside the cell wall, which also includes the cell membrane and other organelles.
Suppose animal cells does not have a cell wall.
Yes.
It has only single boundary, that is the plasma membrane. Then in the case of animal cell, how you can define protoplasm and cytoplasm?
So cytoplasm is the major area in which the organelles are present inside the cell,
Then
and
protoplasm.
protoplasm is the area which also includes the cell membrane along with the cell surface which contains the organelles.
Okay, that means cytoplasm does not include the cell membrane, while
Yes, sir.
protoplasm includes cell membrane,
Yes, sir.
right?
Yes, sir.
Okay, I will explain each and every answers of every questions in the last sessions. Don't worry, don't feel bad. Uh, Vandana, generally
Yes,
we
sir.
study in our class in UG and PG classes. There is a lot of differences in the prokaryotic and eukaryotic cells. Can you suggest me some similarities in prokaryotic and eukaryotic cells which are common?
Similarities in
Yeah.
prokaryote and eukaryotes. Yes, sir. Sir, there is a process called binary fission, which is a reproduction process in the cells, which also occurs in prokaryotes and eukaryotes, which are eukaryotes, especially in bacteria.
Sorry, can you repeat?
And
Can you repeat your statement once again?
so I said the prokaryotes and eukaryotes both have the process of cell division, which may be binary fission, and which is present in prokaryotes and eukaryotes, especially in the bacteria as well.
Okay.
And yes, this is
Okay. So just only one similarities are there in the prokaryotic cells and eukaryotic cells according to you.
no, sir. There are other
Okay.
similarities are also there, and
Vandana, suppose if
yes, sir.
you have a eukaryotic cells and wants to divide it and multiply it, so there will be a process that is called cell division, right?
Yes, sir.
And eukaryotic cells divide by two different kind of a cell division. That is, one is the mitosis and one is another is the meiosis. If
Yeah.
there is a bacterial cells, then which type of a cell division will be there, and what will be the name for that?
Sir, mitosis.
Mitosis.
Yes, sir.
Mitosis is also running in eukaryotic cells and also in prokaryotic cells.
Yes, sir.
Mitosis. Can you say something about the mitosis division? For the mitosis
Sir,
division, there will be a there will be a nucleus and cytoplasm.
yes, sir.
Does bacteria have a nucleus?
No, sir, it's not a well-defined nucleus though.
Then how
In
you can
bacteria.
say that ki bacteria divide by the mitosis cell division? Have you ever studied about it? Yeah, just
Yes, sir, I've studied about it.
uh, okay, okay. Uh, may I ask some questions from biochemistry or biotechnology?
Yes, sir, I'll try to answer.
Okay, Vandana, tell me. We have a different kinds of a biomolecules like as a carbohydrate, protein, lipids, and enzymes, etc. Right? What is the
Yes, sir.
what is the prime function of a lipid in our body?
So lipids are majorly the fat storage molecules.
Lipids are the fat storage molecules.
Yes, sir. Sir, which also provides energy.
Lipids are fat storage molecule. I don't understand this statement. How can I state that is fat is the fat storage molecule? Fat is fat. I am not asking about the tissues, just molecule of that is fat, proteins, carbohydrate.
Yes, sir.
So what is the prime function of lipids in our body?
Sir, lipids are basically. Sir, may I take a moment?
Sorry.
May I take a moment, sir?
Okay.
Yes, sir. So lipids are molecules that act as a chief concentration of in the form of energy, which have about 3.5% of the cell content. And lipids are these are organic substances which are relatively insoluble in water and uh, soluble in uh, organic solvents such as alcohols or ethanol.
Okay. You use a term that is organic molecule. Can you name some inorganic molecules which found in our body?
So I said organic
Yeah. I
molecule.
got it. You said organic. I am asking about the
Yes,
inorganic
sir.
molecules.
So inorganic would be water.
Inorganic means water. Just only water.
Water is there. So blood.
Blood is inorganic molecule.
Yes, sir.
Okay. Some other.
So
Okay. I
these are
uh,
the
I am
main.
giving just a statement. We all have or inorganic molecules without which we cannot survive more than 1 minute. What is this?
Oxygen.
How you can forget oxygen? Life,
Yes. Sorry, sir.
life crucial element. Without oxygen, we can't survive more than 1 minute, and you are just living missing data. How
Sorry,
you
sir.
can? Okay, I can understand your suggestion right now. Vandana, if you have any questions in your mind regarding your interview and regarding your feedback, so you can just ask me and I will suggest, and definitely I will answer all the questions whatever I asked to you. Okay, so now it's your turn, just ask.
Yes, sir.
Yeah.
Sir,
अभी
I
आप
think.
अभी आप कंफर्टेबल फील हो जाइए और आपको कल इंटरव्यू
Yes,
देने के लिए जाना है, तो आप अपने जो भी
yes,
आपके
sir.
डाउट्स हैं, जो भी आपके मन में फियर है, आप उसको एकदम बेहिचक पूछ लीजिए, और ताकि मैं आपको अच्छे से समझा पाऊँ कि क्या चीज़
Yes,
आपको करनी है।
yes, sir.
या
Sir, I think first of all, I would like to analyze myself. Uh,
या
I yeah, I need to work on on my content, my subjective content, especially the basics. I think uh, one thing is that I have studied all this, but I'm not able to recall at the particular moment.
या
And second, I would like to ask you is that as 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 per the previous interviews that the students have given, is what is the differentiation of IPR and the subjective knowledge? So on the basis of the review, I know that I need to work on my subject for sure. And what about the IPR part? Do I need to work more about that? Because you haven't asked me any questions regarding that, so I don't know about that.
Okay, okay, got it. Vandana, whatever you studied during your mains examination preparations in terms of
Yes, sir.
IPR,
Yes, sir.
if you have those materials, just
Yes, sir.
brush up all those IPR material, and there will be
Yes,
no
sir.
need to go in depth or do any extra effort regarding IPR sections. Just
Yes,
keep
sir.
focus and just keep maintain your confidence and concept regarding to the your main subject, especially for immunology. Suppose interviewer will offer to you what is your favorite area, what is your favorite subject, and what is your favorite topic, then definitely you will answer him that is your immunology, right? So there is a need.
Sir, I think I have a doubt in that. I will stick up to a subject which I have more in-depth knowledge rather than immunology. I
So
think
have you any other
immunology is not.
options saying that is you have another area of interest?
Sir, I can say molecular biology.
Molecular biology. Okay,
Yes,
so if you feel that you have also knowledge and concept of the molecular biology and you feel more confident, then definitely there will be an answer. You can say like that, that sir, I am comfortable in molecular biology as well as immunology. It will be better for the interviewer. Matlab means some interviewer will ask the some questions from the molecular biology and from the some immunology. So
yes, sir.
suppose if there will be a 10 to 20 questions from molecular immunology, and round out the you are will able to answer 10 to 12 questions, that will be enough for you. ओके, सो आप एक
Okay,
काम कर
sir.
सकते हो ऐसे, मतलब अगर आपको
Yes,
लगता
sir.
है कि आपका मॉलिक्यूलर बायोलॉजी कंफर्टेबल है, ठीक है एज कम्पेयर टू इम्यूनोलॉजी के, तो आप दोनों को बोल सकते हो। और ये बोलने का मतलब ये हो जाता है कि आपने सामने वाले को इस बात के लिए इनवाइट किया हुआ है कि दैट मीन्स सर
Yes,
आई
sir.
एम रेडी टू
Yes, sir.
आंसर ईच एंड एवरी थिंग्स रिगार्डिंग दैट इज सब्जेक्ट, यू कैन आस्क आस्क सम क्वेश्चंस, ओके। तो ये बोलने का मतलब यही है, और उसके लिए आपको थोड़ा और ज़्यादा पढ़ने की ज़रूरत है वंदना। डेफिनेटली अभी
Yes,
आपने
sir.
रिसेंटली एमएससी किया हुआ है 2023 में, राइट?
Yes, sir.
तो अभी जस्ट आप कॉलेज पास आउट हैं, और हो सकता है कि
Yes,
अभी
sir.
जो जो कॉन्सेप्ट हैं आपने डेफिनेटली आपने पढ़े हुए सारे टर्म्स हैं, दैट इज़ वाई आप
Yes,
यहाँ पे हैं।
sir.
अगर आप पढ़े नहीं होते तो फिर आप मेंस एग्जाम के लिए क्वालिफाई नहीं होते।
Yes, sir.
बट इंटरव्यू में क्या होता है कि सारा जो एक्चुअली आपको ऐसा भी फील हुआ होगा कि सर्टेन टाइम्स के लिए कि आपका माइंड पूरी तरीके से ब्लैंक हो गया।
Yes, sir.
आपको आपको ऐसा लग रहा होगा कि मेरे दिमाग में कोई चीज़ बचा ही नहीं है, कोई कॉन्सेप्ट नहीं है, शायद जो मैं सवाल पूछ रहा हूँ वो आप आंसर नहीं कर पा रहे हैं, ऐसा होता है। उससे
Yes,
अपने आप को बचाने के लिए आपको क्या करना है, जो भी चीज़ें आपको पता है उन्हें आप रिकॉल करते रहिए और उनको थोड़ा-सा अपने माइंड में सेटल डाउन होने दीजिए। जैसे इनिशियली मैंने बहुत सारे आपसे क्वेश्चंस पूछा, जैसे फॉर एग्ज़ाम्पल मैंने आपसे क्वेश्चंस पूछा था साइटोप्लाज़्म और प्रोटोप्लाज़्म में क्या डिफरेंसेस है, राइट? क्या उन क्वेश्चंस को आप अभी मैं ये सवाल जो मैं पूछ रहा हूँ, इस सवाल से मुझे आपके आंसर से मैं ये समझ पाऊँगा कि एक्चुअली में आपका मेंटल कंडीशन कितने स्टेबल है। क्या जितने सवाल मैंने आपसे पूछे उनमें से कुछ सवाल आप बता पाएँगे कि सर आपने क्या-क्या सवाल पूछा है?
sir, the protoplasm and cytoplasm, and you have asked me about the antibody structures and the types of antibodies that are present in human body, and the types of protein structures that are present in humans, and and you have asked me about the plant growth hormones. Plant growth. I'm not. I haven't studied about that concept, so I'm not sure what was that. I think
ओके,
plant growth.
सो स्टार्ट विद द फर्स्ट क्वेश्चंस। मेरा पहला सवाल
Yes,
आपसे ये था कि एंटीबॉडी के स्ट्रक्चर्स को बनाने में विच डिफरेंट काइंड्स ऑफ स्ट्रक्चर्स विल प्ले द रोल, ओके। सो एज यू नो, antibodies are complete structures, okay। Tertiary and quaternary structures are considered as functional state of the any protein in your body. In our body, any protein has to be maintained the tertiary and quaternary structures to perform their functions. Primary and secondary structures are the just preparing of the protein, they are
yes,
not
sir.
the functional stage. That is why I am asking asked to you.
Okay.
And in antibody there are four different structures, that is primary means what, that is the sequence of amino acids.
Yes, sir.
There are the sequence of amino acids, there are folding of that protein, that is secondary structures. they are superfolding of a protein, that is the tertiary structures, and there are four different polypeptide chain interacted with one another, that is called quaternary structures. So on the basis of that data, you can say that for the proper functioning for protein antibody, there are four different level of structures involved in that antibody. That is primary, secondary, tertiary, and quaternary. And
Mhm.
one thing more you
Yes.
have to know, किसी भी ऑर्गेनिजम्स में कोई भी प्रोटीन अगर फंक्शनल स्टेज में है, दैट इज इट, ये वो किस में होगा, टर्सरी स्ट्रक्चर्स में होगा या फिर आपके क्वार्टरनरी में होगा, राइट। दूसरा सवाल मेरा आपसे ये था कि हाइड्रोजन बॉन्ड को आपने लेकर के बताया था कि प्रोटीन्स में
Yes.
मेजॉरिटी आपके हाइड्रोजन बॉन्ड इन्वॉल्व होते हैं, और बाकी आपने एक और बताया था, पेप्टाइड बॉन्ड।
Yes,
पेप्टाइड
sir.
बॉन्ड जो है वो सिर्फ आपका प्राइमरी स्ट्रक्चर इन्वॉल्व होता है, और
Yes, sir.
हाइड्रोजन बॉन्ड जो है वो की बॉन्ड माना जाता है सेकेंडरी स्ट्रक्चर्स का। टर्सरी
Yes, sir.
स्ट्रक्चर्स में डिफरेंट काइंड्स के नॉन कोवेलेंट बॉन्ड इन्वॉल्व होते हैं, लाइक एज हाइड्रोफोबिक, हाइड्रोफिलिक, डाइसल्फाइड बॉन्ड। डाइसल्फाइड बॉन्ड इज ऑल्सो एग्जांपल ऑफ अ कोवेलेंट बॉन्ड, सो
Yes,
एटसेट्रा।
sir.
सो माय क्वेश्चन वाज दैट हाइड्रोजन बॉन्ड को लेकर के, हाइड्रोजन बॉन्ड इज नॉट अ रियल बॉन्ड, इट्स जस्ट इंटरेक्शन बिटवीन द
Mhm.
most electronegative element negative charge and hydrogen positive charge. Have you heard about the partial positive and partial negative charge?
Yes,
Partial
sir.
positive and partial negative, okay. uh, जब कभी भी हाइड्रोजन एलिमेंट, मोस्ट इलेक्ट्रोनेगेटिव एलिमेंट लाइक एज अ फ्लोरीन, ऑक्सीजन एंड नाइट्रोजन,
Yes, sir.
it
Yes, sir.
is only three element in the all periodic table that has the ability to form a hydrogen bond. Whenever hydrogen element will covalently bonded to among these three elements, that is फ्लोरीन, ऑक्सीजन एंड नाइट्रोजन, इनमें से किसी
Yes,
से भी अगर हाइड्रोजन कोई अपना कोवेलेंट बॉन्ड बनाया होगा, तो उसके बीच के मॉलिक्यूल्स में क्या बनेगा, हाइड्रोजन बॉन्ड बनेगा। जैसे आपने
yes,
एच
sir.
का एग्जांपल सुना होगा, हाइड्रोजन फ्लोराइड का। आपने अमोनिया का एग्जांपल सुना होगा, अमोनिया का।
Yes, sir.
वाटर का सुना होगा, वाटर का। तो जहां
Yes,
कहीं
sir.
पे भी हाइड्रोजन कोवलेंट बॉन्ड बनाएगा इन तीनों एलिमेंट में से, लाइक एज ऑक्सीजन, नाइट्रोजन या फिर आपका फ्लोरीन, तो उस मॉलिक्यूल्स में आपका हाइड्रोजन बॉन्ड बनने की पॉसिबिलिटी बनती है। और हाइड्रोजन बॉन्ड इज नॉट अ रियल बॉन्ड, इट इज जस्ट इंटरेक्शन बिटवीन बिटवीन द नेगेटिव एंड पॉजिटिव चार्ज, ओके।
Okay.
इसके बाद मेरा सवाल आपसे था कि आपने टर्म यूज़ किया था ऑर्गेनिक मॉलिक्यूल्स का, तो क्या कोई इनऑर्गेनिक मॉलिक्यूल भी है आपकी बॉडी में? तो डेफिनेटली कार्बन डाइऑक्साइड एंड O2 आर द टू मेजर कंपोनेंट्स ऑफ द इनऑर्गेनिक मॉलिक्यूल, एंड ऑल द salt, whatever the form of the salt present in our body, like the calcium, magnesium, iron, potassium, etc., these all are the example of an inorganic material. You can say that all the electrolytes, nearly all the electrolytes are the chemically salt, and salt are the inorganic molecules. So हमारी बॉडी में ऑर्गेनिक मॉलिक्यूल्स के साथ साथ में इनऑर्गेनिक मॉलिक्यूल्स का भी बहुत बड़ा इंपॉर्टेंट रोल है, और वो इस तरीके से फंक्शंस को परफॉर्म करते हैं। अगला सवाल मेरा आपसे था कि साइटोप्लाज्म और प्रोटोप्लाज्म में क्या डिफरेंसेस है। सो आपने आंसर जो दिया उसमें थोड़ा सा करेक्शन की ज़रूरत है। प्रोटोप्लाज्म का मतलब होता है सेल मेम्ब्रेन के पेरीफेरी में पाए जाने वाले सारे पार्ट, इंक्लूडिंग न्यूक्लियस, न्यूक्लियस के सराउंडिंग का चारों चारों तरफ का एरिया जिसमें आपके सारे ऑर्गेन्स रहते हैं, उसे
Yes.
हम बोलते हैं प्रोटोप्लाज्म। अगर आप न्यूक्लियस को वहां से हटा दीजिए, मीन्स न्यूक्लियस को कंसीडर ना कीजिए, तो न्यूक्लियस के सराउंडिंग का जितना पूरा एरिया बचेगा उसको आप क्या बोलते हैं, दैट इज द साइटोप्लाज्म,
Okay.
ओके।
Sir, I got confused in protoplast and cytoplasm.
Okay, so in mathematical term, you can just state that protoplasm is equal to cytoplasm plus nucleus, while cytoplasm is equal to protoplasm minus nucleus.
Yes.
Got
Okay.
it? I think it will
Yes,
be
sir.
more easy to learn you.
Yes, sir.
ओके। अगर
Definitely.
आप किसी भी सेल्स में, जो कि जिस यूकैरियोटिक सेल्स है, उसमें अगर न्यूक्लियस को कंसीडर अगर आप नहीं करेंगे तो रिमेनिंग जो पार्ट होगा क्या कहलाएगा, साइटोप्लाज्म। जबकि अगर आप न्यूक्लियस को भी कंसिडर करेंगे तो क्या कहलाएगा, प्रोटोप्लाज्म। दैट इज वाई
Okay.
इन द केस ऑफ बैक्टीरिया वी जनरली ऑफर अ टर्म साइटोसॉल,
Yes, sir.
है ना।
Because it doesn't have a well-defined nuclear
या
area.
न्यूक्लियस एज वेल एज साइटोस्केलेटन, bacteria
Yes, sir.
doesn't have a cytoskeleton like structures and doesn't have a nucleus. That is why we also prefer use the term of a cytosol for the material cells. ओके,
Yes, sir.
मैंने सवाल आपसे पूछा था कि एंटीबॉडी के टू पार्ट होते हैं, दैट इज वन इज द एंटीजन बाइंडिंग एंड वन अदर इज द इफेक्टर फंक्शन। मैंने पूछा था कि एंटीबॉडी का रीजन या एंटीजन बाइंडिंग का जो फंक्शन है वो कौन परफॉर्म करता है और इफेक्टर फंक्शन है वो कौन परफॉर्म करता है। सो एंटीबॉडी में दो फ्रेगमेंट है, दैट इज एफ सी रीजन, फ्रेगमेंट ऑफ क्रिस्टलाइजेबल, दूसरा एफ ए बी रीजन, फ्रेगमेंट ऑफ
Yes, sir.
एंटीजन।
Fragment of antibody. Yeah.
सो एंटीबॉडी का जो एंड टर्मिनल है, स्टार्टिंग का जो वेरिएबल रीजन होता है, वो काम करता है कैसा, आपका एंटीबॉडी एंटीजन बाइंडिंग का, और जो आपका सी टर्मिनल है दैट मीन्स कांस्टेंट रीजन है, वो परफॉर्म करता है किसका, दैट इज द इफेक्टर फंक्शन का।
Yes,
ओके
sir. Got it. Yes, sir.
ओके, सवाल मेरा निकला ये था कि डिफरेंट काइंड्स के जो एंटीबॉडी हैं, कितने टाइप्स के डिफरेंट टाइप्स के एंटीबॉडी हैं, और अगर आप एंटीबॉडी का इलेक्ट्रोफोरेसिस चलाते हैं तो कितना बैंड्स आएगा। सिंपली
Yes, sir.
सपोज अगर आपके पास कोई एक एग्जांपल के तौर पे मान लीजिए मैंने आपसे पूछा था दैट इज एंटीबॉडी ए, और आपने बताया कि एंटीबॉडी ए क्या है, डाइवेलेंट है। तो उस डाइवेलेंट को कनेक्ट करने के लिए कि वहाँ पे एक पेप्टाइड चेन पाई जाती है जिसको बोलते हैं जे चेन। क्या बोलते हैं, जे चेन,
JJ.
है ना। जे चेन इज ऑल्सो पेप्टाइड। किसी भी एंटीबॉडी में डिफरेंट फोर टाइप से क्या है, पेप्टाइड चेन है, पॉलीपेप्टाइड। टू उसमें से क्या है, हैवी है, और टू क्या है आपके, लाइट है
Light.
है ना। वो दोनों जो टू लाइट हैं उन दोनों का मॉलिक्यूलर वेट क्या है, सेम है। सो मॉलिक्यूलर वेट सेम होने की वजह से उन दोनों का बैंड कितनी आएगी, सिर्फ एक आएगी। दोनों जो
Yes.
हैवी चेन है उनका भी मॉलिक्यूलर वेट सेम है, इट्स दैट्स व्हाई उनका जो बैंड आएगा कितना आएगा, एक आएगा। तो चार
Okay.
पेप्टाइड चेन को मिला करके बैंड कितनी आई, सिर्फ दो।
Two.
लेकिन चूंकि यहाँ पे
Yes,
सिंस एंटीबॉडी ए हैज एक्स्ट्रा एडिशनल जे चैन दैट इज आल्सो विल बी अ पेप्टाइड, दैट्स व्हाई देयर
peptide.
विल बी एक्स्ट्रा बैंड दैट इज थ्री बैंड।
Three band.
So if we
Yes, sir.
consider antibody A, then there will be a three different band on the electrophoresis gel, and if you consider antibody G, the answer will be two,
Yes, sir.
because antibody G is the monovalent। सिर्फ वहाँ पे कोई
Yes.
जे चेन का कॉन्सेप्ट नहीं है।
Name.
ओके,
Okay. Yes, sir.
और कुछ पूछना है आपको?
So that's all. I need to study
ओके,
more.
वंदना आपके लिए कुछ सजेशंस हैं जिसपे आपको अब तो फिलहाल आपके पास टाइम बहुत कम बचा है। नेक्स्ट डे यू हैव टू
Yes,
गो
sir.
देयर एंड परफॉर्म व्हाटएवर यू हैव लर्न ऑल दोस थिंग्स, बट डेफिनेटली आपके पास आज का टाइम है। प्रेशराइज आपको नहीं होना है, आपको डिमोरलाइज नहीं कर रहे हैं, आपको सिर्फ ये बोल रहे हैं कि आपको अपने सब्जेक्ट पे थोड़ी सी और पकड़ बना लेनी है। Okay, there is no issue in introduction, there is no issue in your communication, and there is no issue in your academic journey. Everything is fine,
Okay.
just you have to focus on your main subject,
Subject.
and
Yes, sir.
just grab the concept that is the terminology, because there will be no need to go in the depth and describe all the detailed mechanism, but you have to answer the terminology, right?
Yes, sir.
So
Basics and the
या
terminology.
बेसिक अंडरस्टैंडिंग मस्ट बी देयर। और तो
Yes,
उसके लिए आपको क्या करना है, अभी सपोज इम्यूनोलॉजी आपने बताया और मॉलिक्यूलर बायोलॉजी बताया, तो कोई अगर आपके
yes,
पास
sir.
नोट्स वगैरह है, कहीं आपने क्लासेस अटेंड किया था मेंस एग्जाम के लिए?
Yes, sir. Sir, कुछ classes लिया था, कुछ actually free lectures, and I have referred to the Pathfinder book and prepared my own notes.
ओके, अगर आपके पास पाथफाइंडर बुक है तो एटलीस्ट आप इतना कर लीजिए कि पाथफाइंडर का जो इम्यूनोलॉजी का जो पोर्शन है उस इम्यूनोलॉजी के
Yes,
पोर्शन
sir.
को एक बार गो थ्रू हो लीजिए, खास करके
Yes, sir.
एंटीबॉडी एंटीजेंस को लेकर के, और
Yes, sir.
हाइपर सेंसिटिविटी को लेकर के, वैक्सीन को लेकर के,
Mm.
ये सारे जो टॉपिक होंगे थोड़ी
Mm.
आपसे सवाल किए जा सकते हैं, तो आप उनको अच्छे से
Okay, sir.
गो थ्रू हो लीजिए। ओके,
Okay, sir.
बाकी और कोई
Okay.
चीज़ का प्रॉब्लम नहीं है वंदना, आपको थोड़ा अपने सब्जेक्ट पे थोड़ी सी कमांड बनानी है, और एक दिन में तो पॉसिबल नहीं हो
Mm.
पाएगा, बट फिर भी जितना आप कर सकते हैं अपनी तरफ से उसको जरूर कीजिए। ओके,
Yes, sir.
चलिए, सो विश यू ऑल दी बेस्ट फॉर योर इंटरव्यू, एंड
Thank you, sir.
कल आप इंटरव्यू जब देकर के आइए तो जरूर बताइए कि कैसा रहा आपका इंटरव्यू, और कैसे
Mm.
टाइप के क्वेश्चन आपसे पूछे गए, एंड डेफिनेटली इफ इट इज पॉसिबल देन वी विल इंटरेक्ट फर्दर for your to know their experience, because there are so many
Yes,
students
sir.
so curious to know the real experiences of the interviewer, so
Yes,
that
sir.
will be good for them. Okay,
Yes, sir. I
Okay.
will definitely do that. Thank you, sir.
Okay. Thank you.
Okay, sir. Thank you.